So science is a process. In this section, we're going to talk about, or you should be able to describe the general process of the scientific method, distinguish between a control group and experimental group in a scientific test, recognize the importance of scientific journals in the reporting of scientific information, interpret information that is presented in a scientific graph, and recognize the importance of statistical analysis to the study of science. And so in this case, we're going to be talking about how science is a process. The role of a scientist is to ask questions about the things that we observe, and the process of science is simply a method for gathering information and acquiring knowledge about the world around us. Now, there isn't necessarily a single formalized set of steps to follow for doing an experiment. However, all scientists generally follow what we call the scientific method. The scientific method is just a way of learning about the natural world by applying simple rules of logic to the way that information is gathered and conclusions are drawn. So the scientific method starts with simple observations about the world around us. However, for a scientist, it is not enough to just observe the world, but you have to ask questions about the things that you're observing. These observations should be prompting a question. So an observation is just a formal way of watching the natural world. It's going to be made with your senses like sight and smell, and then with the help of instruments like microscopes can also be used to make those observations. And so through research, we can make these observations. Now, these observations should then lead us to a hypothesis. So this is the next step, is to develop a testable hypothesis. A hypothesis is just a possible explanation for the observation of that natural event. And we just need to have a direction to travel in. The very best hypotheses are going to be clearly defined statements that we either want to confirm or deny. So think of it as kind of a true-false statement. We don't want an open-ended question, kind of like, or you can also think of it as like a diagnostic test. When we're doing a blood panel, we're not saying, hey, what's wrong with the individual? We break it down into smaller chunks that we can either confirm or deny. When you do a blood panel, you are testing for very specific presences of certain drugs. So you can't just say, hey, I am going to just run like a generic, I'm not going to draw blood and just put it under the microscope and be like, hey, I know exactly what's wrong with you. You're going to have to do specific tests. You're going to have to, let's see, we're going to do a blood count. We're going to look for the presence of specific factors like white blood cells or clotting factors. We are going to test for this presence of specific drugs. So we have to have a lot of yes or no statements. We want to say, hey, is that drug in the bloodstream? Yes or no. If we're doing a STD panel, we're going to test for very specific factors that will tell us whether or not the individual has the STD. We're looking for yes, no, clear, black and white answers. So like I said, a hypothesis is a possible explanation for the observation of a natural event. We're going to use inductive reasoning. That's when somebody uses creative thinking to combine facts into a cohesive whole. Scientists are going to be making a testable prediction based on that hypothesis. So an experiment is going to be using a series of procedures designed to test that hypothesis. It's going to utilize deductive reasoning to make a prediction or expected outcome. And the manner in which the scientist conducts an experiment is called the experimental design. When we are setting up or designing an experiment, there are several key features that all good experiments must have. For example, we're going to need a control. A control is going to go through all of the same steps and the same stages of the procedure, but is going to lack only the one factor being tested. So for example, in class, we'll run if we were meeting in person, we would be running a food testing lab where we're going to be testing different foods for the presences of sugars, starches, proteins, lipids. And what we're going to do is we're going to take like 5 different foods. So I don't know, let's say peanut butter and tuna and rice. I'll just use an example of 3. And we're going to test each one of those for proteins, starches, sugars, and lipids. Now, alongside all of those, we're also going to run water as a control. That water is going to tell us whether or not our experimental results are valid. If we find that protein is found in every single one of those, so the rice tests positive for protein, the tuna tests positive for protein, and the peanut butter tests positive for protein, that's all well and good, but we need to compare that against our water. Our water is our control. If our water also tests positive for protein, then we need to reevaluate our experiment. Potentially there is there's some error somewhere. Maybe there is an error with our protein test. Maybe the dye that we're using is not as sensitive as we need it to be. Potentially our water is contaminated, and so when we're dissolving our protein in water, when we're dissolving our fish in water, potentially the protein that we're detecting is not coming from the food itself, but from the water that we used. So we're going to need a control. The control is going to run through all of the same tests, all of the same procedures, but it's going to be slightly different in that it's going to be containing that one missing variable. In this case, it's water, there's no food in it, and we're going to ensure that our experiment is set up to work the way it's designed. So anyway, that's going to be our control. An independent variable or an experimental variable is going to be the variable that we are testing for. The dependent variable, the responding variable, this is going to be the change or the effect that occurs because of the experimental variable. So those are going to be some of the different characteristics or some of the different components of an experiment. I want to encourage you to spend a little bit of time reading through this section of the text. So results are going to be derived from the experiments. The results could be in the form of data. They might be presented in a variety of formats. You're going to see most often with scientific experiments that we're going to use graphs as tools to help summarize that data. Those could be line graphs, bar graphs, any number of things might be used to help show the significance of that data. So, like I said, data, tables and graphs are going to be two possible formats for the data. These are going to often be analyzed using statistics. Now, statistics are going to be measuring variation. So, things like standard error. Standard error is how far off from the average the data is, or statistical significance is going to be determining how statistically significant are those results, or is the probability that the results are due to chance or some other experimental value. So it's going to kind of help determine how, like, if the change that you're seeing is actually as significant as you are recording or reporting. So statistics are used to interpret data. Statistical data includes standard error, standard deviation to show how uncertain that value is. Statistical significance is determined to evaluate the probability that those results are due to chance. And these studies, once they are finished or can be then published into scientific publications. Now, scientists are going to analyze that data and to reach a conclusion about whether or not the hypothesis is supported. So once you have your data, you then need to analyze your data. You need to then interpret that data, whether or not that data is going to be supporting your original hypothesis. The conclusion of one experiment can then lead to the hypothesis of another experiment, can help us to see, can help branch out from there, further flesh out the information that we are finding. So we're going to actually walk through just sort of an experimental design and look at all the different components. So here we have an initial hypothesis. We have a newly discovered antibiotic B, and we want to compare it to the other antibiotics that are out there on the market. Now, if our question is just, how does antibiotic B compare to other antibiotics out there, that's a very open-ended question. There's a lot of antibiotics out there. How does it compare? That's a really open-ended question. A good hypothesis should be really straightforward with a yes or no response. So what we're going to do is simplify that question down a little bit. We're going to say that antibiotic B is a better treatment specifically for ulcers than antibiotic A. That's the main one that's in current use. So that's going to be our experiment. B is better than A for the treatment of ulcers. So for our experimental design, we're going to use one control group that's going to include subjects with ulcers who are going to be untreated by antibiotics. So here's our control group over here. They have ulcers because it's no good running this experiment on individuals who do not have ulcers. if that is what we're trying to test, is the efficacy of this drug. So the control group does have ulcers, but they are not going to be treated by antibiotics. Then we're going to have 2 test groups that are with subjects with ulcers who are going to be treated with either antibiotic A or antibiotic B. So for this, we're going to need 3 groups total, a control group and 2 test groups. So then we're going to perform the experiment. The groups are going to be treated exactly the same except as noted, meaning that control group is going to receive the placebo, test group 1 is going to receive antibiotic A, and test group 2 is going to receive antibiotic B. In all other cases, they should be treated exactly the same. They should have the same diet, they should have the same exercise regime, they should have the same sleeping habits, they should be given the same drugs in every other category. They should be exactly the same in all instances except for our experimental variable, which would be the placebo versus antibiotic A versus antibiotic B. After a while, or after our experiment has concluded, we are then going to collect our data. An endoscopy, so that's a procedure that allows doctors to examine the linings of throat, stomach, and upper small intestines to check for ulcers, is going to be performed on all subjects. Then the investigators are going to use statistics to determine the effectiveness of various treatments. And on the basis of the data, the investigators are able to conclude whether or not their hypothesis has been supported. And what they found is that when given a control group, we see that roughly 10% of individuals were cured with simply the control. Test group 1 saw 60% cured of their ulcers. However, test group 2, those who received antibiotic B, observed an 80% cure rate. So when we're going to be performing our statistical analysis, it's going to help us determine whether or not B was better or more effective treatment than antibiotic A. And from this data, we are able to conclude that yes, antibiotic B is a better treatment than antibiotic A. So when you're reading or hearing about certain scientific claims, it's essential that you are thinking critically to evaluate the information that you're hearing. You can do this by asking questions like, is the information consistent with information from other sources? Gather information from as many sources as possible. The thing is that if antibiotic B really is a better treatment for ulcers than antibiotic A, then this should be true every time in every situation with multiple tests. Maybe we should see that if we maybe change sleep and we give, maybe our patients who are taking antibiotic A versus antibiotic B, maybe we will submit everybody to having varying amounts of sleep. So maybe with less sleep, antibiotic B, we'll see the effectiveness of antibiotic B or the antibiotic A. So what we should see is that under multiple conditions, under multiple experiments, we should see consistency in our results. And that's something that's very, very common in the scientific community. When somebody publishes their results, when somebody publishes their scientific findings, it is up to their peer group to validate that information by performing either the same experiment or other experiments that are going to corroborate those results. So you want to be able to gather information from as many sources as possible. If there is only one cluster of data out there, if there is only one experiment that has ever been performed that says, hey, this new treatment out there is 100% effective, but there's only ever been one test performed, it's not a highly scientific process. It's not highly verified scientific data. So you want to be able to gather information from as many sources as possible. Don't passively accept information as true just because it sounds good. Do your research. You should be looking at how reliable is this information? What is that source of information? Is it possible that the research is biased? Who stands to gain if you accept that information as true? Was this information obtained scientifically, or do they rely on anecdotal evidence or scientific research? And were those results interpreted correctly? That's something that's going to become hugely important this semester as we are performing our labs. I'm going to place a big emphasis not so much on the accuracy of your data collection, but the accuracy of your data interpretation. You should understand your data and what it means. And so that's going to be a big focus this semester. All right, and that is it for section 1.3.